This patient is on intravenous fluid containing glucose, as he is not in a condition to take nutrition orally. Why does intravenous fluid supply glucose, whereas the food we eat contains such carbohydrates as starch and glycogen? The food we eat enters the alimentary canal, where it undergoes mechanical and chemical breakdown. Only digested nutrients are absorbed into the blood. Digestion is the process of enzymatic breakdown of complex nutrients in the food, such as complex carbohydrates, into simple absorbable forms such as glucose. This is the reason why intravenous fluid can slide into the blood. As we know, carbohydrates are not the only macronutrient. There are other macronutrients, namely proteins and fats. Let's see how each of these nutrient types is digested in our body. Common food items such as bread, cereals, potato, beans and bananas are rich sources of carbohydrates. This activity, when you next eat a piece of plain bread will start tasting sweet. Can you guess the reason for this? The bland taste of the bread is because of the starch present in it. When we chew the piece of bread, it gets mixed with saliva. Saliva is produced by the salivary glands. Saliva contains an enzyme called salivary amylase or ptyalin. This enzyme breaks down starch to maltose, a form of sugar. It is this maltose that gives the sweet taste. What if we don't chew our food well? Will the remaining undigested carbohydrates in our food not be digested at all? There is no carbohydrate digestion in the stomach, as the gastric juice does not contain any carbohydrate digesting enzyme. But carbohydrate digestion continues in the duodenum. Here, the pancreatic amylase present in the pancreatic juice acts on starch and converts it to maltose and other sugars. The epithelial lining of the small intestine secretes intestinal juice. It has several enzymes that further break down maltose, sucrose and lactose to glucose and fructose. These simple sugars formed after breakdown of the complex carbohydrates are then absorbed into the blood. Food items such as butter, cream, cheese, egg yolk, nuts and oils are rich sources of fats or lipids. No fat is digested in the buccal cavity. As the food reaches the stomach, only a very small amount of fat is digested to fatty acids and glycerol. This process is catalyzed by a weak gastric lipase enzyme. Chyme, an acidic mixture of partially digested food and gastric juice, now enters the duodenal region of the small intestine. Here, bile secreted from the liver and stored in the gallbladder is added to the chyme. Bile emulsifies the fat molecules. Bile acts as a detergent and breaks large water-insoluble fat globules into smaller particles. Emulsification helps enzymes to act on fat molecules. Pancreatic lipase, an enzyme secreted by the pancreas, breaks fats into fatty acids and glycerol. Fatty acids and glycerols are absorbed by the lymphatic vessels present in the villi region. Can bile be considered an enzyme? Bile is not an enzyme. It only makes it easier for the lipase enzyme to act on fat molecules. Milk, cheese, meat, eggs, and pulses are rich sources of protein. The digestion of proteins begins in the stomach. Enzymes that digest proteins are called proteases. The acidic condition of the stomach helps in the conversion of pepsinogen to the active enzyme pepsin. Pepsin breaks down complex proteins into peptones. Peptones are smaller protein molecules. The cells of the inner stomach lining contain proteins. What prevents pepsin from destroying the cells of the stomach wall? Mucus, secreted by specialized stomach lining cells, forms a layer on the stomach lining, thereby preventing pepsin from destroying it. The smaller protein molecules formed in the stomach now move to the duodenum. The process of protein digestion continues in the small intestine. Peptones are further broken down to smaller peptide molecules by the enzymes trypsin and chymotrypsin, secreted by the pancreas. These smaller peptides are further broken down to amino acids by peptidases, enzymes that are present in pancreatic as well as intestinal juice. These amino acids are finally absorbed